Welcome back, everyone. So we've made it to the end of the semester. We are in chapter 20 now, and we've really saved the best for last. We are talking about the regulation of gene expression. So what it is that allows some cells to turn on genes and to turn off other genes. All the cells in your body have all the same DNA, but they're not all acting the same because they have different genes turned on. There's a lot of really interesting stuff in this chapter that we just don't have time to discuss it all. So please make sure that you do read your textbook for this chapter because it really is an interesting one and one that is applicable to a lot of current research going on in science. So everything in nature has to be regulated. It's an important aspect of everything. And gene expression is no exception to that. By selecting what genes are turned on and turned off, or expressed or not expressed, a cell is able to be more efficient. It's only making the proteins that it needs for its cell type or for its situation, and it's not making proteins that are unnecessary. It's not wasting energy on things it doesn't need. And although we really do focus on mammals in this class, a lot of really important understanding of gene regulation came from bacteria. And so we're going to spend a bit of time talking about operons, which are exclusive to bacteria, but also very interesting in how they're able to control the genes that they express depending on the situation that they find themselves in. Some genes are constitutive genes. That means that these genes are expressed all the time. Whether there's a lot of sugar or not a lot of sugar, a lot of ATP, not a lot of ATP, whether there's lots of cells growing or you're not growing, constitutive genes are always there. I think of things like ribosomes. You're going to need ribosomal RNA and ribosomal proteins for pretty much anything you're doing. You're going to need genes to, make your mitochondrial components and other organelles pretty much all the time. So those constitutive genes are always on. For a lot of the genes, their gene expression is regulated, and only those genes are expressed only when your cell needs them. So regulated genes are for enzymes that the cell does not constantly need. One way that we regulate the amount of enzymes there is by starting and stopping transcription. So when your cell needs it, you turn it on, you turn on transcription and start. And when your cell no longer needs it, you stop transcription. So that's adaptive enzyme synthesis. We only make the enzyme when we need it. Bacteria use different approaches for regulating enzyme synthesis, whether it's catabolic or anabolic. So a catabolic enzyme, something that degrades a sugar, we're going to talk about the Lac Operon. Your bacteria will only express those genes when there is lactose to be digested. It doesn't make those genes if lactose isn't around. There are also anabolic pathways or synthetic pathways, things that your cell needs to make. We're going to look at the tryptophan operon, and that is something that is only turned on when it's not present. So the absence of tryptophan will turn on this pathway, because if we have it already, we don't need to keep making more. So catabolic enzymes exist just to degrade specific substrates. When we're talking about lactose digestion, the catabolic pathway that degrades lactose, this disaccharide, into monosaccharides, one of the central steps is this enzyme beta-galactosidase that breaks this disaccharide into the two monosaccharides, galactose and glucose. But even before that can happen, you have to get lactose into the cell. And the way that you transport lactose into the cell is through an enzyme called galactosidase permease. So galactosidase permease or beta-galactosidase is an enzyme you only need when lactose is present. You don't need to be making a lot of this here when you don't have lactose. So we only make beta-galactosidase when lactose is present. So we're only going to turn it on or induce it when lactose is there. So that's an example of substrate induction. So without lactose, these genes are not going to be expressed. But with lactose, we're going to pull off the repressor and allow these genes to be expressed. So when the gene is turned off, it's repressed without lactose. And so this is an inducible enzyme. It will be created and regulated in an inducible way. For anabolic pathways, the amount of the enzyme that you produce by a cell is usually going to be correlating inversely with the concentration. So when we have rising levels of tryptophan, you're going to start repressing the production of tryptophan enzymes. So that's end product repression. So that's going to be working kind of the opposite direction from how our lactose pathway works, right? So lactose, we only make the genes to digest it when lactose is there. For anabolic pathways, we only build things when we don't have it. We build the things we need. So we're looking at things from two different perspectives, and please don't get those two different kinds of pathways confused. So in this feedback inhibition with our tryptophan pathway, when you have molecules of enzyme, they're still present, but their catalytic activity can be inhibited. So we're looking at protein synthesis and not just protein activity here. Okay, so the lactose operon, the Lac Operon is an inducible operon. It's turned off unless it's induced. It's like run-of-the-mill, everyday type of setting is for it to be turned off. And a big part of understanding the lactose operon was the discovery of two types of genes. The first were the genes that coded enzymes used to bring lactose uptake and metabolism. And the second was the regulatory genes that control the activity of those first group, those genes that actually create the enzymes to digest lactose. So there's 3 lac genes, lacZ, lacY, and lacA, and they belong to the single regulatory unit. They all share the same operator and the same promoter. So this is called an operon. An operon is a group of genes. They all have related functions, so they're all related to the digestion or the catabolism of lactose. They're clustered together and they'll be turned on and off together. So the one promoter will turn them all on, the one repressor will keep them all turned off. This is much less common in eukaryotes than it is in prokaryotes. So of these enzymes, lacZ encodes beta-galactosidase, which is what hydrolyzes lactose into monosaccharides. lacY encodes galactosidase permease, which is involved in bringing lactose into the cell. And lacA gene encodes transacetylase. That adds an acetyl group to lactose as it enters the cell. So in this operon, we have genes with related functions. They're clustered together. So their transcription is going to be regulated and coordinated together. So for the whole operon to induce their transcription, we have an additional gene, the lacY gene. So the lacY gene stands for inducer. So the lacY gene is going to make protein products that regulate the expression of the Z, the Y, and the A gene. So the regulatory gene product will inhibit the expression of other genes. So here lacI is a repressor protein. So the Lac Operon has these 3 genes we just talked about, Z, Y, and A, preceded by an operator and a promoter. And then before that is the regulatory gene, the lacI gene. And this lacI gene regulates the Lac Operon. Transcription of the operon will begin at the promoter. It'll go through the operator and it'll transcribe the lacZ, lacY, and lacA gene. So you'll have one mRNA transcript with all 3 genes on it. So that is a polycistronic mRNA. The interaction between the operator and the repressor protein is an important part of how the Lac Operon is controlled. So the lacI gene is going to create a Lac repressor. This gene is outside of the Lac Operon. And the repressor will bind to the operator. So when this repressor is bound, RNA polymerase can't transcribe these genes. It just is a physical block, prevents RNA polymerase from turning these genes on. So when that inhibitor, when that lacI repressor is bound, the whole Lac Operon is turned off. To turn on the gene, the inducer molecules, so here when we're talking about lactose, allolactose will have to bind to the repressor. When allolactose binds to the repressor, it changes its shape. So now this repressor is not bound to the operator. So when lactose is present, it binds to the repressor and takes it off of the promoter. So now the RNA polymerase is able to start transcribing these genes. So when lactose is not present, the repressor is bound to the operator, genes are not turned on. Lactose is present, lactose binds to the repressor doesn't bind to DNA anymore, RNA polymerase can bind to DNA and start transcribing those genes. So this repressor is an allosteric protein. Allosteric refers to having two different shapes, two different conformational states. Whether it has the effector on it, it'll be in this conformation, not bound to DNA, or if it does not have the effector on it, it'll be in this conformation, bound to DNA. And so it's able to change its shape to control RNA transcription. So the effector for the repressor is allolactose. Allolactose is an isomer of lactose. Once lactose comes into the cell, it can be isomerized into allolactose and turn on the genes. The binding of allolactose to the repressor will induce a conformational change. That then means this repressor is incapable of binding to the operator. So lactose triggers the induction of the enzymes that are coded by the Lac Operon. Once allolactose is bound to the repressor, now you're able to start making beta-galactosidase, galactoside permease, and transacetylase. There's one more layer in this control, though, and that is having a catabolite activator protein, or a CAP. So if lactose is around as the only food source, then your bacteria are going to use lactose as a sugar source. However, if lactose is around and glucose is around too, your cells get a little picky, and those bacteria are going to choose to use glucose instead. They will ignore that lactose as long as glucose is around, because glucose is the preferred carbon source for all of these cells. So catabolite repression is how bacteria will make sure that glucose is used first, keeps that lactose operon turned off when glucose is there. So in catabolite repression, the effector molecule is cyclic AMP. Here's cyclic AMP. So glucose will inhibit adenylyl cyclase. So the more glucose that is present, the less cyclic AMP is made. So if you have lots of cyclic AMP, because your glucose level is low, it will attach to CAP and bind DNA and help the RNA polymerase bind to the operator and turn on the lactose digesting genes. If glucose levels are high, then we don't have as much cyclic AMP here anymore. So CAP is not bound to cyclic AMP. If CAP is not bound to cyclic AMP, CAP doesn't bind to DNA. So it's not bound to DNA, and transcription of this Lac Operon is only going to happen at a low level. So we have two layers of control here, whether or not lactose is present to allow this to be transcribed, and whether or not glucose is present, which determines if this operon is being transcribed at a low level or at a high level. The CAP binding site is upstream of the promoter in the Lac Operon. So here are your lacZ, Y, and A genes coming down, your operator, your promoter, and then here's your CAP binding site. There's similar sites found in inducible operons for metabolism of other sugars. So when glucose is around, they're not going to be used as a carbon source as long as glucose is there. The binding of the activated cyclic AMP bound CAP will enhance the binding of the RNA polymerase to the promoter, which will promote the operon transcription when glucose is absent. So we make high levels of this Lac Operon when glucose levels are low and lactose levels are high. If lactose isn't there, we're not going to turn it on at all. But if glucose is there, it's still only being transcribed at low levels. So the Lac Operon is an example of dual control of gene expression. It's subject to both negative and positive control. It is under control by glucose concentration and under control by lactose concentration. Most tightly regulated genes in eukaryotes and prokaryotes are going to have tight dual control for just the right situations for that gene to be expressed. Factors whose binding of DNA inhibit transcription are negative regulators, and those that enhance transcription are positive regulators. Okay, so we talked about how we control the genes that help catabolize sugar. Next, we're going to talk about how we control the gene that helps with anabolism of tryptophan, how we turn on the expression of tryptophan so we make enough tryptophan when it's not present, and we stop making it when we have plenty around. All right, I'll see you later. Bye.